Welcome, online listeners and viewers, and thank you for joining this Science AAAS webinar, Immune Profiling of Single Cells, a new milestone in multiomic biomarker analysis. My name is Jackie Oberst, and I'm the Assistant Editor for Custom Publishing here at Science. I'll be moderating today's webinar. Our guest speaker will be Dr. Swati Acharya from Stanford University in Palo Alto, California, whom we will learn more about shortly. Before we get started, I'd like to provide the lay of the digital land for our online viewers. At the top right of your screen, you'll find a photo of today's speaker and a View Bio link, which you can click on to read more details about her background and research. Underneath the slide viewer is the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to BD for sponsoring today's webinar. So let's dive in, shall we? The ability to simultaneously interrogate protein and mRNA information in thousands of single cells is a recent development, allowing greater insight into the biology of different disease states. Powerful tools such as antibody oligonucleotide conjugates, AbSeq, and RNA sequencing, RNA-seq, allow researchers to gain a more comprehensive understanding of the intricacies of immune-related diseases, thus providing opportunities for more personalized treatments. During this webinar, our expert speaker will describe how the power of multiomic single-cell analysis was applied in recent asthma research, in which thousands of T cells were captured using oligoconjugated cell surface antibodies and interrogated using targeting sequencing to identify immune cell biomarkers. A digital gene expression profile of thousands of individual cells was generated in a single experiment without the need for robotics or automation, enabling identification of distinct cell surface proteins and hundreds of gene expression biomarkers. Without further ado, I'm very pleased to introduce our speaker to you now, Dr. Swati Acharya from Stanford University. Dr. Acharya works as a scientist at the NADU laboratory at Stanford University, where she applies her knowledge of immunology, genetics, and mechanistic pathways of immune modulation and T cell development to identify molecular mechanisms and biomarkers of impaired regulatory T cells and type 2 T helper cells in asthma and allergy. She investigates clinical samples from twin cohorts using cutting-edge omics such as AbSeq, or assay for transposase-accessible chromatin using sequencing, single-cell transcriptomics, and CyTOF-based proteomics, together with targeted single-cell and plasma mass spectrometry and other novel methods. Welcome, Dr. Acharya, and thank you for joining us today.
Thank you, Dr. Oberst, and hello, everyone. Today I'm going to speak about a common airway disease called asthma. Asthma has been on the rise with a need to understand and outline the disease mechanisms for development of precision biomarkers for various endotypes of asthma that will aid in diagnosis and allocation of therapeutics. According to the latest World Health Organization estimates, as of December 2016, approximately 235 million people worldwide had asthma. The characteristics of asthma are described by the Global Initiative for Asthma, GINA, as a heterogeneous disease with a complex genetic trait characterized by varying levels of bronchoconstriction, airway hyperresponsiveness, smooth muscle hypertrophy, structural airway remodeling, mucus secretion, and chronic inflammation. Airway inflammation is mediated by excessive memory responses to inhaled allergens, along with comorbidities that include multiple organ allergies such as allergic rhinitis, conjunctivitis, atopic dermatitis, and food allergy. Non-allergic disorders such as obesity, gastroesophageal reflux, and psychiatric conditions are also implicated. Cluster and other non-hierarchical analyses have identified subtypes of asthma associated with differing causal pathways, natural histories, and responses to interventions. Asthma can be categorized into two broad phenotypic groups as shown here, T2 type asthma and non-T2 type asthma. The T2 type inflammation occurs in over 80% of the children and in the majority of adults with asthma in association with allergen sensitization and atopic dermatitis. Asthma features might begin in utero and is associated postnatally with viral-induced wheezing and allergen sensitization. Strong genetic, environmental, and epigenetic mechanisms continue into childhood asthma. Asthma is more common in boys than girls in early childhood, throughout puberty, and into early adulthood. However, boys experience asthma remission at a higher rate than in girls. In adulthood, asthma can spontaneously remit or begin de novo, and the factors that lead to the emergence and regression of asthma, irrespective of age, are poorly understood. Let's look at the immunologic and pathobiological features of severe asthma. Mechanistic pathways of immunological and pathological features on the type 2 inflammation are shown at the top right, and the lower right figures depict how antigen induces TSLP stimulation, differentiation of Th2 and IL-2 cells, and release of type 2 cytokines, i.e. IL-4, IL-5, and IL-13. Activation of mast cells, smooth muscle contraction, eosinophilic infiltration, remodeling and amplification of inflammatory cascades, i.e. IL-3, IL-4, IL-5, and IL-9 follow, along with production of IgE through the action of IL-4, activation and recruitment of eosinophils through IL-5. IL-3 induces smooth muscles to induce hyperresponsiveness, remodeling and stimulation of the epithelium for cytokine and mucus production. Mast cells also synthesize prostaglandin D2, which stimulates the upstream cells and eosinophils through its action at CRTH2. The non-type 2 inflammation includes factors such as infectious agents and irritants that stimulate the innate immune system via IL-33 and ST2, IL-25 via IL-17RB, which stimulate the IL-C2 and Th2 cells. Activated Th17 and the products stimulate the neutrophils. These mechanistic pathways facilitate inflammation of the air passages that results in temporary narrowing of the airways that carry oxygen to the lungs, as was shown in the lower right figure in the previous slide. Current treatments that are available for asthma can be categorized into two groups, i.e. small molecules and biologics. The small molecules include short-acting beta agonists, inhaled corticosteroids, long-acting beta agonists, ultra-long-acting beta agonists, leukotriene antagonists, and the antisense oligonucleotide treatments. Biologics include the very successful monoclonal antibody against IgE, omalizumab, and interrogation of pathways for new drug development include IL-5 pathway with a binder for IL-5, IL-4 and IL-13 pathway that include the AMG 317 and the famous dupilumab, which are both IL-4 alpha blockers, and lebrikizumab, which is the IL-13 blocker. The IL-17 pathway has also been harnessed for binding the IL-7 alpha receptor for asthma drugs. When we talk about asthma phenotypes and endotypes and inflammatory signatures, we have to keep in mind that the determinants of pediatric clusters do not mirror those in adults. Broadly, we can classify asthma phenotypes into 5 groups: the adults with severe asthma, early onset of allergic disease with the active Th2 inflammatory pathway, increased airway eosinophils, mast cells, IgE, and pheno-nitric oxide. Group 2 includes the adult onset of asthma with early eosinophilia, absence of allergic disease, active Th2 pathway, and IL-4, IL-5, and IL-13 in the blood. Group 3 symptoms are mainly exercise-induced and increased mast cells. The group 4 have minimal Th2 response and notable obesity. The group 5 folks shows sputum neutrophilia and Th17 cells. The common biomarkers that are looked at while testing serum, blood, sputum are IgE, eosinophil, neutrophil, surrogate periostin, and DPP4, and there are biologics associated with it that are used for treatment, which is shown in the last column. I left a gap in the slide to remind us that there is a paucity of endotype-specific biomarkers in the field of asthma research in our era of precision medicine. Efforts to resolve precision biomarker signatures for various asthma endotypes or mechanisms require involvement of research using multiomics platforms at a single-cell level with increased accuracy and sensitivity, which can develop into point-of-care testing using non-invasive devices, hopefully soon. Ongoing research and development in various labs all across the world are sure enough making this possible. So keeping in mind a multiomics platform, looking at RNA and protein together are important as their complex relationship and regulation shape the heterogeneity in biological samples. If this complex RNA-protein relationship does not exist, and RNA expression is proportional to the protein expression, then the correlation would be high, like this theoretical graph on the left. As shown in this review noted in this slide, The reality is that the correlation of mRNA and protein are not always high. In this sample, the R-squared is 0.41. Therefore, there is a need to bring solutions that help researchers delineate both mRNA and protein expression, ideally in the same sample. In order to study the complexity of gene regulation, researchers have employed many approaches to understand RNA and protein expression in cells. In today's workflow, I'm going to talk about my favorite sample, the PBMC, and we do a lot of cycle in the lab to obtain, and from which we obtain the PBMCs, and we sort or use different flow cytometry methods to interrogate these cells. Traditionally, bulk samples are divided to detect gene expression, either using Western blot for protein or qPCR for RNA. While these bulk tools have made significant strides towards understanding gene expression, there is an increased push to understand cell-to-cell heterogeneity within the biological sample. Single-cell analysis tools such as flow cytometry and cell sorting have propelled the field of research forward to understand cell-to-cell heterogeneity. With the emergence of single-cell RNA-seq, this tool complements flow cytometry well to understand the whole picture of the single cells in both RNA and the protein field. To profile proteins using a modern flow cytometer, 8 to 10 different colors can easily be measured in one sample. The most advanced cytometers can even measure up to 18 channels at once. Using CyTOF, cytometry by time of flight, a high-performance mass cytometer like the Helios enables the analysis of more than over 45 markers using metal-conjugated antibodies and uniquely allow their quantitative determination with negligible spectral overlap, a result of exquisite resolution between mass detection channels. Advances in single-cell transcriptomics using droplet or gem-based methods such as InDrop, DropSeq, and 10x genomics to analyze transcriptomes on a cell-by-cell basis through the use of microfluidic partitioning to capture single cells and perform barcoded next-gen sequencing has led to the development of combinatorial single-cell proteomics and RNA-seq. As shown in the far right, a few such notable multiomics platforms are AbSeq, or antibody-based sequencing, CytSeq, cellular indexing of transcriptomes and epitope sequencing, ReapSeq, RNA expression and protein sequencing, AbSeq or aptamer and transcriptome sequencing. Due to lack of time and space, I've not mentioned a whole bunch of novel transcriptomic and protein combinatorial sequencing methods, which also include studying methylation and proteome together, methylation and RNA-seq together, and so on. And each of these major significant sequencing methods have also been developed into look at cells in a more unique way. Let's start with AbSeq. It was first developed by the Acharya lab at UCSF. Here, the cells are stained with antibodies and labeled with unique sequence tags, as seen in A. To read out single-cell protein expression, a microfluidic workflow conjugates the antibody tag sequence bound to the cell in B and unique cell barcode sequence in C via splicing by overlap extension PCR, as is shown in D. This is performed on over 10,000 single cells in parallel, and chimeric products are pooled and sequenced. To obtain single-cell protein information, the data is sorted by barcode. Unique molecular identifiers are included to correct tag counts due to duplicated sequences resulting from PCR bias during sequencing library preparation. We have adopted this method for our research besides other methods, and we've used the BD platform. The BD antibody oligos have a DNA attached to the antibody that has a poly-A tail, which can act as an RNA mimic to be used in BD Rhapsody system. This poly-A tail would be recognized by our cell capture bead, just like how mRNAs would. A unique antibody-specific barcode is used to recognize the specific antibody instead of reading out fluorophore channel information of an antibody in flow cytometry. This barcode is sequenced to delineate what protein is expressed in a specific cell. The PCR handle in the 5' end of the antibody allows efficient library generation for sequencing. Lastly, a long antibody-specific barcode allows users to count how many antibodies are captured and sequenced in a single cell level. The BD Rhapsody cell capture beads are designed uniquely in a way that various levels of barcoding are baked in. First, the cell label, CL, is a barcode unique to the bead itself, so one can track which sequencing read is from which cell. Second, the unique molecular identifier, or UMI, on the bead allows one to track which sequencing read is from which RNA transcript. When the mRNA is captured, the cDNA synthesis is performed to hard code the transcriptome information into the bead with the various levels of barcoding. This bead design is magnetic and does not melt, so one can store a subsample of these beads for later experimentation. CytSeq uses the DNA barcoded antibodies to convert detection of proteins into a quantitative sequenceable readout. Antibody-bound oligos act as a synthetic transcripts that are captured during most large-scale oligo-DT-based single-cell RNA-seq library preparation protocols, such as used in 10x genomics, DropSeq, or DDSeq. It's very exciting to see from the website, site.com, that there are new developments where one of the more important ones are cell hashing, where oligo-tagged antibodies against ubiquitously expressed surface proteins are labeled uniquely in the cells from distinct samples, and this gives a multiplexing capacity a whole new turn. There's also the ExciteSeq, which is the expandable CRISPR-compatible cellular indexing of transcriptomes and epitopes by sequencing for a high-throughput characterization of transcriptome, immune receptor clonotypes, surface markers, sample identity, and sgRNAs, guide RNAs in each cell. Then comes along ReadSeq, which is the RNA expression and protein sequencing. For ReadSeq, cells are labeled with antibodies conjugated to DNA barcodes instead of with traditional antibodies conjugated to fluorophores. These barcodes consist of 65 to 66 base pair DNA divided into an NGS read, which is used as a primer for amplification and sequencing. A unique 8 base pair antibody barcode and a 24 to 25 base pair poly-D sequence. These antibody barcodes labeled cells are then subjected to single-cell RNA-seq using the chromium single-cell 3' solution from 10x genomics. Paired and sequencing reads for mRNA and protein are generated, and protein sequences aligned using an antibody dictionary that associates each antibody to its unique 8 base pair barcode. Then cells are grouped by their chromium-generated cell barcodes, resulting in a digital protein and a signature and a gene expression matrix. One of the newer ones is AbSeq. AbSeq mirrors AbSeq and its successor, cellular indexing of transcriptomes, or CytSeq, and the RNA expression and protein sequencing, or ReadSeq. In that, it allows combined surface and transcriptome profiling for tens of thousands of single cells at a high throughput base. Like antibodies, aptamers are affinity probes capable of specific binding to target epitopes, and aptamer binding can be multiplex. The principle of AbSeq is as follows. In the first section of the slide, A heterogeneous cell sample is incubated with diverse aptamer library containing a poly-A sequence on its 3' end. Cells expressing epitopes of interest are decorated by corresponding aptamers in the library, and non-binding aptamers are washed away. Single cells of the washed cells suspended are co-encapsulated with beads carrying unique DNA barcode in a microfluidic device. Each droplet contains lysis solutions to lyse the cells. Aptamers and mRNA molecules can hybridize with the barcoding beads by means of their poly-A sequence. Using the barcode beads as a primer in reverse transcription and DNA polymerase reactions, the droplet-specific unique barcode is fused to an mRNA aptamer, providing a cell-specific identifier. Pulling all the beads after barcode fusion, sequencing their contents in parallel, and deconvoluting the aptamers and the mRNAs allow evolution of epitope profiles in the single cells. Since the cell-specific barcode is shared between the aptamers and transcripts, the epitope data can be combined with the single-cell transcriptome for further interdependent analysis. This brings us to the need for multiomics and combinatorial methods to decipher biomarkers of asthma endotypes, the problem that I started this session with. The ability to simultaneously integrate protein and mRNA information in thousands of single cells is a recent development allowing greater insight into the biology of different disease states such as asthma. Here in our research, we attempted to combine measurement of specific proteins and gene transcripts in individual purified CD4 T cells from peripheral blood of monozygotic twins with discordant asthma status in order to understand the role of cellular diversity in disease development. As I mentioned earlier, we used the BD Rhapsody-based AbSeq platform along with our multiomics protocol to understand and define the endotype biomarkers of asthma. The BD Rhapsody system is very self-contained. BD Rhapsody cartridges are optically clear, as shown here, which is an advantage to allow visualization and quantitation of cell bead pair events in a well format. You can also see the green color, which is because of the dye that's added. Here is the actual picture of the BD Rhapsody. Beads are paired with the cells just prior to lysis. The right picture shows how the cell capture beads have an oligo-DT capture site to grab the poly-A transcripts. And also the antibody tags are also captured on the BD Rhapsody cell capture bead. The BD Viewer platform allows the users to visualize and analyze their own single-cell RNA-seq experiment conducted across multiple platforms. Fortunately for people like me, no command line usage is required. Simultaneous detection of mRNA and protein levels in single cell using any of the protein mRNA sequencing methods we discussed. To understand whether the BD AbSeq would affect RNA expression on the BD Rhapsody platform, cells that were treated with or without BD AbSeq antibodies were analyzed and then subjected to parallel library preparation, sequencing, and analysis. The expression correlation of mRNA targets did not show noticeable differences between RNA profile and with and without BD AbSeq. Expression profile in individual cells using the t-SNE showed overlap of gene expression profiles, suggesting there is no detectable difference in the mRNA expression when using BD AbSeq in the single-cell platform. Data generated from a 40-parameter BD AbSeq analysis with mRNA on BD Rhapsody was analyzed using t-SNE. What was seen using mRNA only is that while major cell types are distinguishable, such as monocytes in green, some cell types, such as CD4 and CD8 or an orange circle, can be close in mRNA expression profile. When BD antibody-seq data generated using a t-SNE plot-focused plot was added into generating a t-SNE plot, while it changes its relative position within the plot, distinct CD4 and CD8 clusters were observed, and 2 clusters within the cell type were revealed, suggesting increased granularity of data in a single-cell sequencing experiment using AbSeq panels. Moreover, delineation of expression of differences between mRNA and protein of the same gene were observed. For example, a popular cell marker, CD4, actually has low expression of mRNA, as shown here, whereas the protein levels in the T cells is extremely high. Bringing in the protein information using BD AbSeq allowed a deeper understanding of their expression differences, as shown here. To analyze gene expression changes using the BD AbSeq on BD Rhapsody during dynamic cell response, a 4-hour stimulation of PBMCs using PMA inomycin, followed by assay for gene expression changes for both protein and RNA levels were performed. Aliquots of cells were treated with a transcription inhibitor, a translational inhibitor, and a proteasome inhibitor alongside DMSO as control to test whether BD AbSeq can provide insight in a well-used immunological model system using multiomics platform. Between 0 and 4-hour time points using multiomics upregulation of known activation markers, that is CD69 using protein and TNF using mRNA, reveals useful data. By looking at the drug response, we saw that the CD69 upregulation was largely due to rapid protein translation because adding a cyclohexamide rescue is upregulation. On the right side, the TNF mRNA is starting to get transcribed, supposedly by the fact that transcription was attenuated using the actinomycin D. Furthermore, single-cell studies showed gene regulation heterogeneity within the same sample cells gated for T and B cells reveal differences in the mechanism of CD69 upregulation. On the left, in T cells, CD69 upregulation can be perturbed by addition of cyclohexamide, suggesting a strict protein translation effect. In B cells, the CD69 upregulation can be perturbed by addition of both actinomycin D and cyclohexamide, suggesting that this gene is regulated both by transcription and translation. This type of tool opens a wide variety of understanding of heterogeneous biological samples. Research studies involving twins can help distinguish between the effects of genetics and environment, and they have significantly contributed to the information we have on biological traits and diseases. We investigated differential gene expression profiles for asthma using PBMCs from monozygotic twins discordant for the disease, i.e. asthma. We used the BD Rhapsody and BD AbSeq platform, as I said earlier. We researched the shared memory T cell signatures among twins with asthma as well as divergence from their healthy siblings. To do this, we adopted the BD AbSeq platform, wherein single cells associated with barcoded magnetic beads in individual wells were lysed to hybridize mRNA and antibody oligo onto the bead, retrieved, and then cDNA was synthesized. The final products were sequenced and analyzed. Clustering analysis was performed using DataView software, as I described earlier, and which is shown on the lower left. The lower middle figure shows the clustering of each of the 5 twin pairs that cluster together. The Disney plot on the far right shows the differences in gene expression between healthy, that is red, and asthmatic, which is blue, in the discordant twins. Differentially expressed candidates were revealed using this AbSeq strategy in peripheral CD4 T cells of twins discordant for asthma. In this assay, we use the AbSeq, which with a combination of targeted panels instead of doing the entire transcript sequencing, which gave us benefit of cost bias, as well as we wanted to target specific genes and look at their expression levels using the AbSeq method. Future directions. I think precision medicine in the management of asthma in clinical practice requires detection of valid molecular biomarkers that can be linked to point-of-care biomarkers. The use of unbiased approach to clustering of various omics platforms, genomics, transcriptomics, proteomics, and metabolomics as a promising tool to determine composite biomarkers of asthma phenotype, endotypes, and personalized medicine. Predicting biomarker-based signatures of response to biologic therapeutics of asthma, and understanding the heterogeneity of asthma to predict therapeutic response towards development of composite biomarkers from blood, urine, exhale, breath to develop appropriate in-practice diagnosis. These should be the future, I think, the future directions to take this field into a multiomic strategy and define specifically the mechanistic pathways involved that can categorize endotypes of asthma. I would like to acknowledge the Sean Parker Center for Allergy and Asthma Research for providing me the opportunity for research, my mentor, Kari Nadeau, and people in the lab that were involved in the project, Shu Chen, Diane, Tara, Wenming, and Rebecca. The Chang Lab has been extremely helpful in this collaboration. I thank Dr. Howard Chang, Dr. Ansuman Swati, and Yuning Wei. The Human Monitoring Core with Holden Macker and Jehua Ji, as well as the Stanford Functional Genomic Facility, John Kohler and Dhananjay Wong, and a lot of thanks to the folks at BD, Abby, Shireen, Jessica, and Christine for helping us. We would definitely want to thank our funding sources at the National Institute of Allergy and Infectious Diseases, as well as the Heart, Lung, and Blood Institute at NIH. And here's a picture of our Sean Parker Center team, both the clinic and the lab, wet lab folks. Thank you very much.
All right. Thank you, Dr. Acharya. So it just remains for me now to thank today's speaker, Dr. Swati Acharya from Stanford University. Please go to the URL right now at the bottom of your slide viewer to learn more about the resources related to today's discussion, and look out for more webinars from Science, available at webinar.sciencemag.org. This webinar will be made available to view again as an on-demand presentation within approximately 48 hours from now. We're interested to know your thoughts about this webinar. Send us an email to the address now up in your slide viewer, webinar@AAAS.org. Again, thank you to our speaker and to BD for their kind sponsorship of today's educational seminar. Goodbye.